Okay, so today we're talking about cell junctions and cell signaling. The reason we're talking about these together is basically we want to get an idea of how cells work in community, right? Humans, this is not going to come as a surprise to you. We're more than one cell, right? We're multicellular organisms. A lot of the organisms that we see in the world are multicellular organisms. We almost never see single cellular organisms unless we're in a biology class. So understanding some of the components of cells that allow them to interact with each other and work as a group is really important to understanding the purpose of cells in the world. Okay. So we're going to start with cell junctions, which are basically how cells connect to each other. Okay. Before we do that, I want to talk about something called the extracellular matrix. So that should ding, right? This is one of the things that you are supposed to draw in your problem set. So what the extracellular matrix is basically material kind of like our cytoskeleton, but outside the cell. So the extracellular matrix is this network of proteins and polysaccharides, so proteins and carbohydrates, that are made by the cells and then sent to the external environment. They kind of surround them in a layer. The purposes of this is kind of like the purpose of our cytoskeleton, right? They're going to provide strength and structural. Ooh, sorry about my audio, guys. Maybe I can turn some captions on. Let's see. Ooh. Are words appearing? Okay, I'll try to talk slowly and hopefully the captions at the bottom will help. Okay, so the extracellular matrix is basically like the cytoskeleton, but outside the cell. So it's made by the cell, but it doesn't stay inside. It gets transported to the outside. So it has roles that are similar to the cytoskeleton. It provides strength, provides structural support. So it again has this scaffolding kind of effect, just like our cytoskeleton. And this also allows it to organize tissues, put them in layers, and just like the cytoskeleton allows things to move along it, the extracellular matrix is also going to allow cell signals to kind of move along it. Okay, so here we see a layer of cells in beige, and then in yellow, these kind of yellow tubey looking things. Those are the protein fibers in our extracellular matrix. And then we can see the polysaccharides in kind of purple and green. So there are lots of different components here. Sometimes we'll see cells embedded within the extracellular matrix. How cells are organized and how they're interspersed throughout is going to be dependent upon different tissue types, which we'll talk about in A&P. Okay, so now that we have an idea of the extracellular matrix, we're going to move on to the junctions themselves. So I'm going to clarify here what we're looking at. This red tube way at the bottom of the image, that's a blood vessel. Then this blue layer, we're going to think of as our extracellular matrix. So some proteins and stuff, not within the cell, but originally created by the cell. Then in beige, we have the cell itself. The basal part of the cell is the part that's facing the bloodstream, so that bottom part of the cell. The apical side of the cell is the top. So in this case, it's the part with all those basically squiggly bits, right? Those projections at the top, that's the apical part of this cell. So when we say cell junctions, we're thinking about the things that connect cells to each other. So two neighboring cells to each other. We can see some of our blue lines and some purple lines connecting cells to each other, some yellow lines. We are also going to have cell junctions that connect the inside of the cell to the extracellular matrix, so to that supportive structure outside the cell. Okay. So our first type of junction is an anchoring junction. And what you want to immediately word associate in your mind with anchoring junction is the cytoskeleton. So the anchoring junction is for structural support. That's basically its job, is to create shape and hold it there. So it's going to physically, mechanically attach the cytoskeleton, which is that scaffolding inside the cell, either right to the cytoskeleton of the neighboring cell or to the extracellular matrix, which are these proteins that are outside the cell. So if you see something attaching to the cytoskeleton, we're thinking anchoring junction. So there are proteins that allow these different cytoskeletons to attach either to each other or to the extracellular matrix. You may learn about what these proteins are called and how they function in the future, but we're not going to worry about those right now. We're just thinking cytoskeleton physically attached to another structural kind of protein. Actually, I'm going to annotate just real quick. I'm going to show us where we can see an anchoring junction here. So we would think of our anchoring junction. We think of like these blue lines as being part of the cytoskeleton, right? So when these blue lines are connecting to blue lines in the neighboring cell, we would identify this portion, right, as an anchoring junction because we've connected cytoskeleton to cytoskeleton. And then these red lines are also another type of depiction of cytoskeleton. The cytoskeleton has many parts. So if this is our cytoskeleton and we see this junction directly with the extracellular matrix, this blue layer, that's also an anchoring junction. So those are the anchoring junctions we can see in this picture. So anything else that looks like that as well would be an anchoring junction, right? Here, here, over here, connecting to the other neighboring cell. Those are all anchoring junctions. Our second type of junction is a tight junction. So a tight junction is basically when we have two cell membranes sewn together with a big long protein fiber. So tight junctions, their job is to create like a seal that molecules can't pass. And this is important because it's going to force anything that wants to get from the apical side of the cell down to the blood on the basal side of the cell to actually go through the membranes of that cell, which means it's going to force all those molecules to be regulated by our different transport proteins in those cell membranes. So anywhere where we don't want leaking through a layer of cells, we're going to expect to see tight junctions. So like in the future, you'll talk about epithelial cells, which are like your skin, but they also line internal cavities. You're going to expect to see tight junctions there because lining cavities is really important that we have a good seal surrounding them. I didn't mention about the anchoring junctions, but you might expect anchoring junctions and a lot of extracellular matrix in places like bone, for example, right? The places that are about structure and mechanical properties. Okay, so if we're looking at this picture, we're looking for our tight junctions. We're going to expect to see places where the membrane is being held really close together, pressed against a neighboring membrane, and where we see threads physically just kind of sewing up the membrane. So that's here, this top one is a depiction of a tight junction where we have physically sewed the membrane of two neighboring cells together using some protein fibers. Our gap junctions are actually really important to how a lot of cells function, how cell signals get past certain types of cell signals. So you will hear about gap junctions in the future as well. But the basic idea of a gap junction is that it connects the cytoplasm in one cell to the cytoplasm in the next cell. So it's a big hole, unlike a channel protein, right? Like it's actually made of like 8 proteins in this kind of donut-y shape. So this pore between is really big. The purpose of this gap junction is that anything that's inside the cytoplasm here can just flow directly into the cytoplasm of the next cell. So they're still 2 cells physically separate, but in terms of their internal context, they can almost behave kind of like the same cell, because any molecules, any chemicals that are in one cell are just going to be able to flow freely into the next cell. So they're kind of going to be in equilibrium with each other. Whatever's in one is in its neighbor. So yeah, so it's made of connexons, proteins, looks like little donuts. So if we were looking for it in our big picture on the right that has all the types of junctions, right? So we're looking for where we see tunnels, connecting membranes, connecting cytoplasm to cytoplasm. So here are connexons and our little tunnels, our gap junctions here at the bottom. These donutty guys. Okay, so that is it for our 3 types of cell junctions. They do have subtypes, which you may talk about way in the future, but that's good for now. That's what you need to know for now, and that should get you where you need to be in your future courses. Brings us to cell communication, aka signaling. So cells communicate by making signals, detecting signals, responding to signals. When we talk about a signal, we're talking about a molecule. So a signaling molecule could be like a neurotransmitter, but we'll also have cells like detecting glucose in the environment. So what exactly the signal is gonna be different. But our signaling molecule is always going to be out in the environment somewhere, right? It's going to bind in to a protein in the membrane, right? So this is an integral transmembrane protein, right? Integral because it goes into this nonpolar fatty acid layer in the center here, transmembrane because it has part outside, an extracellular part, and a part inside the intracellular part. Okay, so when the signaling molecule binds into this protein, one of the main things we know about proteins is that they have shape. When the signaling molecule binds into the protein, it changes shape. So if you compare this tail end, this intracellular portion here versus here, we can see that the shape is different. So we call that activating the receptor. So without a signal, it's inactive. Once the signal binds in and it changes shape, it's activated. As it activates this receptor, that then sends off a signaling cascade. So just like, kind of like dominoes, right? We have this whole pathway, all sorts of chemical processes that happen as a result of this receptor being activated. We call these molecules that are made in the middle of this like pathway. So our dominoes that are falling to eventually create a cellular response, we call these second messengers. So second messengers are like signals inside the cell, right? Our signaling molecule in most cases is staying outside the cell. So we have second messengers inside the cell passing along this signal. Okay. So receptor act in steps, we're going to go through numbered steps, but they're pretty simple. So the first thing is we have receptor activation. The signaling molecule binds into the receptor gets activated. We can call this a conformational change. Conformation really just means shape. So conformational change is a shape change. Second thing that happens is we have signal transduction via second messengers. So these other molecules pass the signal into the cell. The third thing we have happen is a cellular response. So this is often going to happen in the nucleus, right? Often what second messengers are going to do is get up to the nucleus and then tell molecules in our nucleus basically to read different genes in different parts of DNA in order to change something about the cell. So it may change what enzymes are in the cell and in what concentrations. It may tell the genome to read genes that are going to change something about structural proteins, aka our cytoskeleton, which could alter cell shape or movement, or it could alter transcription factors, which would have kind of a cascade of effects on other genes that are read in the genome itself. So this signaling pathway is going to have an effect here. It doesn't always have to go to the nucleus. Sometimes there will be a direct effect on an enzyme or a direct effect on a structural protein. That can happen too. So according to our learning objectives, and these are the most important ones, we're going to talk about 3 types of signaling. So the first type is paracrine signaling. So what's happening in paracrine signaling is we start with an original cell. So we're going to think about our cell sending the signal as this beige one, right? This one is our signaling cell, the one sending out the signal. So it creates this molecule, right? It sends it out through exocytosis, right? And if this molecule happens to be a protein, we traced how it would be made and folded and packaged and exported by our different organelles. So it makes this signal, sends it out into the environment. And in paracrine signaling, what then happens is the signal runs into a receptor in a nearby cell. So not the same original cell, not a cell far away, a cell close by, like a neighboring cell. So any of these yellow cells could be receiving the signal. And then we have that whole pathway that we just talked about. The receptor gets activated, there are second messengers, and then there's an eventual response. So the cell that receives the signal is our target cell. The cell originating the signal is our signaling cell. In autocrine signaling, the first part of this is going to look kind of the same. So in autocrine signaling, we still have our original cell here in the middle. So our originating cell, just gonna star it, this one. So it's making this signal and it's sending it out to the local environment. The special thing that happens in autocrine signaling is that while that signal can still go and affect other neighboring cells, right? The original cell itself, the signaling cell, has the same receptors that all those neighboring cells do. So the signal like loops back. Auto means like self, right? So an autocrine signal is one that affects the original cell itself. So in an autocrine signal, we'd expect the same cell that is creating the signal to also respond to the signal that it just sent. Our final type of signaling is endocrine signaling. So what differentiates endocrine signaling is that our target cells are really far away. So we still have an original cell, this beige one over on the left. It's sending out a signal, but that signal has to get into the bloodstream, and then it's going to travel way far away to its target cell. So you could think of it as like a signal originating in a heart cell, then having to get into your bloodstream and reach somewhere in your liver or something like that. That would be an endocrine signal. And really, when we're thinking endocrine signaling, we're going to refer to those signals that are sent through the bloodstream as hormones. Like that's what a hormone is it's a signal sent through the bloodstream somewhere far away. So our receptor is going to be in this faraway cell, our faraway target cell, somewhere else. That's it. So we are going to move to those problem sets in our group. And basically, you're just going to draw on one of each of these types of junctions, and then you're going to follow the directions to draw a signal, both as it's produced and received. And then you're going to identify what type of signaling you just drew. So pay careful attention to which cell is supposed to be producing the signal, which cell has the receptor. It's in the directions, and then you should be able to look at your image and figure out whether you were drawing autocrine, paracrine, or endocrine signal.